Hi everyone. This video introduces our MobiCom work, PDLens, done in collaboration with the University of Rochester Medical Center. Parkinson's disease is a brain disorder that will affect 930,000 people in the USA by 2020 and is estimated to affect 1,238,000 people by 2030. The NIH reports that Parkinson's disease has four main symptoms, which are tremor, stiffness of the limbs and trunk, slowness of movement, and impaired balance. Although there is no cure for Parkinson's disease, there are medicines and other therapies that can often relieve some symptoms. In general, drugs prescribed for PD have three functions, which are to increase the level of dopamine in the brain, affect levels of other brain chemicals in the body, and help control non-motor symptoms. Here is an example to show how medication helps people with Parkinson's disease. In this video, the subject is asked to do the following activities, which are standing up from a chair, walking unassisted in a straight line, and coming back to the chair. The video on the left shows the patient's performance before medication, and the video on the right shows the patient's performance after medication. We find that our patient does much better after taking medication. However, around 3 to 4 hours after a dose of levodopa, which is a common Parkinson's disease drug, symptoms re-emerge or worsen. Then, symptoms typically improve 15 to 45 minutes after taking another dose of levodopa. This phenomenon is called wearing off. Wearing off tends to happen more frequently as Parkinson's disease progresses, and when it does, the control of both motor and non-motor symptoms fluctuates. As wearing off becomes more frequent, it may become increasingly difficult to control symptoms and predict when medications will be effective. So our goal is to identify drug resistance in time, thereby adjusting drug forms to improve the symptoms. An occasional clinical visit cannot let a physician fully understand the progress of Parkinson's disease. The in-home self-assessment sometimes doesn't work because the process of drug resistance is so slow that people may not notice it in time. This is an effect called creeping normality. In this paper, we present PDLens, a smartphone-based end-to-end system to facilitate self-detection of Parkinson's disease drug effectiveness. It leverages the built-in smartphone sensors to collect digital PD biomarkers from users' daily activities, which are sensitive to dopamine levels in brains, and therefore respond to drug intake. We propose a ranking-based multi-view neural network for the drug effectiveness detector. Convolutional layers and LSTM layers are adopted for feature extraction. Then we use RankNet to compare the symptom severity at two different times, I and J. Weights in the neural network are not shared among each view, but they are shared between the two branches of time I and J. We collaborate with the University of Rochester Medical Center to enroll 81 participants with Parkinson's disease. This clinical trial lasts for 6 months. Participants are asked to perform 3 activities in each day, which are voice, walking, and standing. For the evaluation of drug response detection, our goal is to measure the performance to differentiate between before and after drug intake. For each user, we use an 80-20 train test split. This confusion matrix chart shows the results of drug response detection. Our system achieves an accuracy of 91%. Also, the recall is close to precision, suggesting that we can differentiate 2 stages without obvious bias. The figure on the right shows the correlation between the score in standard drug effectiveness test and that in PDLens. Although all the patients respond to drugs, the drug effectiveness varies from person to person, where the highest score is 0.72 and the lowest score is 0.2. Moreover, the score predicted by PDLens is correlated to that in standard drug effectiveness tests with a Pearson correlation coefficient of negative 0.36. This result meets our expectation that a participant with poorer drug will correspondingly receive a lower score in PDLens. For more information, Please see the full 20-minute video and read our paper. Thank you.